All right, everybody. Good afternoon, good evening. It's Dr. Kaz here, and I am going to be doing our Chapter 10 session review here. And so let's get right down to it. Okay, we're going to be discussing muscle tissue. And as you know, there are three types of muscle tissue that we learned about in Chapter 5 when we were studying tissues. There's skeletal muscle, cardiac, and smooth. And if you believe it or not, that this muscle tissue, all three muscle tissue types, will make up almost half of your entire weight. So that's really, really important to kind of understand that when we talk about muscle tissue. Yes, the muscles themselves are considered the organs of this system here. Okay, but half of your body weight, pretty impressive, is going to be made up of this muscle tissue. As you know, as you've been studying for lab for chapter 11, all the different muscles, it seems like there's hundreds. Well, you guessed it right. There are close to 700 different skeletal muscles in our muscle system. So keep that in mind. Now, thank goodness we don't have you learn all of those, but it's quite an endeavor. And we're still discovering new muscles. I won't say every day, but frequently. I believe it was in 1996 where we discovered the suboccipital muscles. We recently, a few years ago, discovered another muscle in the thigh. So again, if you think that anatomy is not dynamic, you're incorrect. It is dynamic. It is changing. We are still learning new things. So let's get into the skeletal muscle and talk about the functions. Now remember, muscle moves things. So that the first function of skeletal muscle is to move the body. And obviously we know that skeletal muscle attaches onto the bones. But not only that, it can attach onto other connective tissues. All right, but what it does is when it attaches onto the bones, it allows movement, ambulation. Maybe you've taken a drink of water, as I'm about to do. We'll see it with nonverbal communication all the time through facial expressions. It allows us to eat, chew, swallow, breathe. Okay, so think of skeletal muscle with body movements. Maintenance of posture. Okay, as I am sitting here talking at my computer, I am sitting upright. Well, in order for me to do that, all right, I have skeletal muscles that are firing right now and contracting to keep me upright. And so when you are doing certain poses, like for example, yoga, you would be utilizing these muscles too to help stabilize the joints as you're going through those movements, but also to maintain or hold that body position there. Protection and support, okay, does help to hold some of the internal organs in place. Especially down when we get into the abdominal region there, you don't have the luxury of the ribs holding everything in. So what we'll see is, all right, in that scenario, we have muscles in some areas there. Now there's the story of Harry Houdini, who died because he allowed somebody to punch him in the abdomen. He was convinced that he was strong enough, or at least his core muscles were strong enough to provide him with quite a bit of protection. And so he would quite frequently challenge people to punch him in the stomach to show them just how strong he is or was. And so unfortunately, this one time he was caught off guard and the gentleman punched him and hit him in the spleen, caused his spleen to rupture. And the crazy part was he allowed the man to do it again, all right? Because he said he wasn't ready the first time. So we'll also see some of the functions for skeletal muscle and elimination of materials. As we're moving things through our body, okay, we will see that eventually when we get into the GI tract or the digestive system, that things are going to move from one opening to another opening, from mouth to anus, we call it. All right, it's essentially just one big long tube, all right, with a couple different modifications along the way. And what we'll see is when the contents make it to the end or the exit there, right, we have, for the most part, voluntary control over their elimination. This is called potty training, right, or bathroom or toilet training. So when you were younger, your parents were teaching you how to voluntarily eliminate wastes, right, both through bowel and bladder usage. They were actually teaching you how to control these skeletal muscles that acted as sphincters. Heat production, we talked about that in chapter 1. Right, as one of the results with temperature regulation, if your body temperature starts to drop down, right, one of the responses through negative feedback is to cause the skeletal muscles to shiver, contract and relax very, very quickly, and that will generate heat. So after talking about functions, we have to move into some of the characteristics of skeletal muscle. And if you notice here on our list of characteristics for skeletal muscle, the first two characteristics of skeletal muscle are very similar to the first two characteristics of nervous tissue, right? So with skeletal muscle, we have excitability. Its ability to respond to a stimulus, all right, that will eventually cause a contraction. But how we go about this is similar to how we went about it with the nervous system. We are going to change that membrane potential. You remember that difference in charge across the plasma membrane of the neuron? Well, we're going to see something similar to that when we talk about skeletal muscle tissue. Conductivity, again, right? We are going to be utilizing electrical charge and the movement of an electrical charge for this stimulation and this excitability. So we actually have to propagate this electrical charge and have it travel down a cell membrane. And so we're going to see how that works in the skeletal muscle. Contractility, right? This tissue has the ability to change length. Right? We'll talk about the sliding filament theory here on how you have these filaments, all right, these small little protein fibers that will slide past each other. And when they do that, it causes the movement that you'll see when a muscle is contracted. Extensibility, well, you've heard of the saying, what goes up must come down. Well, when we contract a muscle, we are then going to have to relax the muscle and then allow it to return to its original length. But what about stretching our muscles, right? You've heard about stretching muscles because you've probably done it, okay? So when we talk about the extensibility of a muscle, it's how much that this cell, this tissue can be stretched, excuse me, okay? So we're gonna actually lengthen this tissue and lengthen the cells of this tissue. And then finally, we have elasticity. Right, that is basically the recoilability, or once we've contracted the muscle or if we've stretched it, its ability to return back to its original length. Kind of like the elastic waistband that you might have on your athletic shorts there. So very, very similar, right, in that regard. Okay, so let's move on to, all right, what it is that we're going to be talking about this chapter. In this chapter, we're going to be talking about skeletal muscle primarily. We'll touch on cardiac and smooth. We're really going to focus a lot on skeletal muscle. So, like we did in chapter 7, when we were talking about the bones were the organ. Well, in this chapter, in chapter 10, our skeletal muscle is going to be an organ. We're going to see that there's going to be several different types of muscle, or not muscle, excuse me, different types of tissue that are going to work together when we're discussing the physiology or the function of skeletal muscle. So a few different definitions here, right? A muscle fiber, okay, is going to be, all right, where we have several of these muscle cells, right, that are going to be wrapped together, right, in which we call a fascicle. So we'll bundle these muscle fibers together into a bundle, and we will call that a fascicle. So our muscle itself is made up of several fascicles, right? And so these fascicles are made up of these muscle fibers, which we can also call myofibers. Right. So when you hear me say the term muscle fiber, myofiber, I am talking about one single skeletal muscle cell. And so we can see that here in our picture. Here we have our one single muscle fiber. Right. This is the skeletal muscle tissue here. And then we bundle several of those together, and we're going to get our fascicle. You can see we have a bunch of fascicles. And then we bundle all the fascicles together, we'll get our entire muscle fiber. Muscle. And so we're going to talk about, we saw this before, how in our nerves we have these 3 connective tissue coverings. Well, in our muscles, we also have, all right, these 3 connective tissue coverings. All right, so since we're on the topic of connective tissue, let's talk about a couple different types of connective tissue. Tendon, which is a dense regular connective tissue, and an aponeurosis, which is also a dense connective tissue. Think of an aponeurosis like a thin sheet, a thin tendon here. So when we talk about tendons and aponeuroses, we're going to talk about these structures that are going to connect muscle to something. Okay, it could be muscle to bone, it could be muscle to skin, it could be muscle to connective tissue. So we use these tendons and aponeuroses to do this. All right, then we have this other type of connective tissue called deep fascia. Now there's superficial fascia and there's deep fascia. You can figure out what the names mean, okay? Deep is going to be, all right, well below the superficial. All right, so we're going to see that with the deep fascia, this is going to be the fascia that if you ever do a gross dissection, you have to separate certain muscles, like in the quadriceps in your thigh there, okay? The deep fascia will actually separate individual muscles from one another. All right, it is a dense irregular connective tissue. And it is, I might as well go over the three different, I didn't review the three different types, jumping all over the place here. All right, here's the three different, all right, connective tissue coverings. We have the endomyosin, that is the deepest one, the innermost layer, okay, and that is going to surround each individual muscle fiber. Then when we bundle those muscle fibers together into fascicles, then we have our perimyosin, and that surrounds the fascicle, and that's going to be made up of a dense irregular connective tissue. And the epimyosin covers the entirety of the muscle, and that too is a dense irregular connective tissue. Right. The endomyosin is a loose connective tissue. So we want to have that loose connective tissue, all right, because we're going to be loading this area up with tons of blood vessels and whatnot. All right, and then finally, our other type of connective tissue is the superficial fascia. Right, and if you look here, all right, the superficial fascia is made up of areolar and adipose connective tissue. That looks similar. Those ingredients look similar to the hypodermis, okay? Well, the superficial fascia is another name for the hypodermis. It will be deep to the integument, underneath the skin there, right? And so this layer is what we will actually see when you peel back the skin, right, during a gross dissection, right? This is the layer that you'll see after peeling back the skin. This is the superficial fascia, and that separates the muscles from the skin underneath. All right, of course, we can't go anywhere without blood vessels and nerves. And so, right, skeletal muscle is going to be highly vascularized, obviously. Okay, muscle does a lot of work, right? And if it does a lot of work, it's going to require a lot of energy. So it has a high metabolism. So we need to make sure that we can deliver the nutrients, any of the respiratory gases, to and from that tissue there. Right. Also, skeletal muscle tissue is innervated by nerves, specifically by somatic neurons. All right, so if you remember from chapter 12, when we were talking about the different types of neurons, all right, we had, when we were talking about the functional portion of our nervous system, we had our somatic nervous system, which was the voluntary or consciously controlled portion here. So that's what we're going to see here. The somatic neurons are the ones that we can actually voluntarily and consciously control. So that allows us to have voluntary control over muscle contraction for skeletal muscle. All right, so let's talk about the parts of the muscle cell, our myofiber here. Okay, so we're going to rename some of the structures. We did that before in chapter 12, okay, when we were talking about the nerves. And so we'll do some of that here. Okay, all cells have cytoplasm, except when we're talking about the skeletal muscle, we change the name of the cytoplasm to sarcoplasm. All right, it's going to have all the other types of organelles in there, but when you hear the term sarcoplasm, you'll immediately know that we're referring to, all right, the cytoplasm inside of our muscle cell. All right, we learned way back when, all right, some of the characteristics of skeletal muscle cells, and we notice that they're long and cylindrical, but they'll have multiple nuclei. They're multinucleated, and I want to say I have I don't have that picture on the slide. All right, no big deal. All right, but basically, right, during our embryological development, right, we have these myoblasts. Right? Those are the primitive, right, stem cells, and what will happen is these cells will fuse together during our muscle development period. And so they retain their nuclei, and that is why we'll see, all right, how muscle cells will be multinucleated. And then those that don't fuse together, okay, they'll stay close by and they'll differentiate into satellite cells. And of course, satellite cells are going to be the support crew. Right? They're going to help to provide structure support, nourishment to this tissue, but also if there's damage to these cells, they'll help to repair the damage. All right, just like any other cell, we have a plasma membrane, except for our muscle cell here, we're going to call it the sarcolemma. Remember the name of the plasma membrane that we gave to the axon in the neuron, we call that the axolemma. Well, here we're going to call our plasma membrane the sarcolemma. So we have a new structure here in the sarcolemma. It's called the T-tubules, okay? Stands for transverse tubules. If you've ever seen when you take a bath and you're letting the water out of the bathtub, and as the water gets close to being completely empty, you'll see this little kind of like whirlpool form over the drain there, right? That's what the T-tubules remind me of, right? They're these invaginations that go deep into the cell, and they kind of look like that little whirlpool that you would see as the water's draining out of the tub. That's what it reminds me of. Well, what we'll see here along the sarcolemma is we're going to have, right, some transport proteins. And so we learned about the different types of transport proteins before when we talked about the neuron. Well, in this scenario with our sarcolemma, okay, and our T-tubules, we have voltage-gated ion channels, similar to what we saw in the neurons. And so these voltage-gated ion channels, we know that they allow the diffusion of ions across the plasma membrane, across the sarcolemma. And so this will help to facilitate and propagate the conduction of electrical signals, because that's what we're going to need to generate in order to stimulate this muscle. All right, so we have our voltage-gated ion channels, and we'll have something new here. Right, we've got these special kind of transport proteins called voltage-sensitive calcium channels. Now, voltage-sensitive calcium channels are very specific. Obviously, the stimulus is going to be, we need some sort of electrical change in the sarcolemma to trigger the opening of these channels. But when they open, it'll allow calcium to move through the channel. And that's going to occur once an action potential has been generated, all right, way back where the nerve actually is going to synapse on the actual muscle cell. All right, we'll get into all that fun stuff here in a little bit. So here we can see, all right, the structure and organization of our skeletal muscle fiber. Let me kind of zoom in here. So you can see what we've done. Okay, here's our complete muscle. Then we have all these little fascicles. We just yanked one fascicle out, and now we're pulling out the muscle fiber. Now we're going to take a closer look at our muscle fiber. And this is what we're seeing. All right, our muscle fiber, okay, is going to be made up of all these tube-like structures. And these tube-like structures are called myofibrils. And so you can see, right, within these myofibrils, we'll have some dark regions alternating with light regions. This is what gives us, right, the striated appearance to skeletal muscle. But there's several other structures that we're going to see. You're going to see there's nuclei all over the place, multinucleated. We've got mitochondria all over the place, the powerhouse of the cell. Okay, so they're going to generate ATP for us so we can contract our muscle. But you see these bluish green tubes all over the place, and then you can see like this web-like tube structure on the outside of the myofibrils. We're going to talk about those. All right, so if you notice here, all right, in the myofibrils, in between the myofibrils, right, you'll have mitochondria, like I said before, some nuclei, some other organelles, but also we have our sarcoplasm. Okay, so in between these big tube-like structures, or actually they're really tiny, right, these myofibrils, they just sit, all right, and they live inside the sarcoplasm. Okay, that's going to be important here later on, so keep that in mind. All right, then, right, we wrap the whole thing up, all these myofibrils up in our plasma membrane, our sarcolemma here. And you can see we have all these holes throughout the sarcolemma, and those are the openings to the T tubules. Okay, you can see them all over the place. And it's tough to see here, okay, but you have the green here, all right, that represents the T tubules. So if we zoom in closer to one individual myofibril, right, you can see it a little bit better. So I want you to think of this whole kind of structure here like someone wearing a long sleeve shirt. So here's your arm, and then the long sleeve shirt represents, all right, the sarcoplasmic reticulum, which are these blue tube-like structures, and that includes the T tubule and this other structure called the terminal cisterna. We'll get into that in a moment. Okay, so the arm is going to be the myofilaments here, not the myofilaments, the myofibrils. And then the shirt sleeve will be like the sarcoplasmic reticulum, this tubular network that sits on top of that. So we're going to get into now, right, some of the purposes of these structures here. So you can see here when we're talking about our sarcolemma, right, here's the plasma membrane or our sarcolemma. You see there's a charge across it, positive, negative. We have our voltage gated channels here, right? And here's a couple familiar faces: voltage gated sodium channel, voltage gated potassium channel. Same scenario. They're going to allow the diffusion of ions, okay, across the sarcolemma. And then you can see how, all right, we get this invagination here. Whoops, that's the T tubule. And then we have these specialized channels. These are the voltage sensitive calcium channels. And we're going to talk about their function here in a moment. They're pretty impressive, I got to tell you. It's really kind of neat. So let's talk about that. All right, folks, let's talk about now some of the parts here of our muscle cell. Okay, so to do that, all right, we're going to start talking about the myofibrils. Okay, the myofibrils are what I was just showing you, those long tube-like structures that are inside the cell itself, right? So we'll see several thousands of them, right? And these myofibrils are going to be encased or covered by the sarcoplasmic reticulum. And so we're going to break this down here for you here. So what exactly is the sarcoplasmic reticulum? We'll use that analogy. That's the shirt sleeve that I was telling you about, but it's a tubular structure. Similarly, you know how we were seeing the endoplasmic reticulum? It was that tube-like structure that was close to the nucleus there in chapter 4, and we've talked about it where protein synthesis occurs. Right. So our sarcoplasmic reticulum is going to be on the outside of these myofibrils. And so think of it like that shirt sleeve. And so it has all these tubes. We're going to get into the actual purpose of it. Well, at the end of the sarcoplasmic reticulum, we have this structure called the terminal cisterna. And so I think of the terminal cisterna like a shirt cuff or a hem. So normally when I'm teaching this, I'm in class and I always wear a shirt and tie to class, long sleeve shirt, and I can demonstrate this a little bit easier. So you'll just have to rely on my words on this one. So this tube-like structure system that you're seeing here, okay, this is our sarcoplasmic reticulum, all these blue tubes. At the end here, see these blue tubes on either end of all that sarcoplasmic reticulum? That's the terminal cisterna. So this is, again, that's the shirt sleeve, and then at the end, this is like the cuff or the hem there. And you'll notice that right next to it is this green structure, and that's the T tubule that I talked to you about. And then on the other side of that, we've got another terminal cisterna that's attached to another tubular network, another sarcoplasmic reticulum. And so this structure here is called the triad. It's made up of two terminal cisterna and a T tubule. So that's what we're referring to when we're discussing that. But what really is important about the terminal cisterna is that's where we keep, all right, calcium. Okay, calcium is a very important ion for many different physiological structures, but for this chapter, for chapter 10, it is crucial for us to have calcium so we can initiate, all right, a contraction. So we can stimulate a muscle to contract. Think, and I'm going to liken calcium to like that princess that's trapped in the tower. Right? We need to go and rescue that princess. We need to liberate her. We need to let that princess out. So right now, all right, the terminal cisterna is going to contain a bunch of this calcium that we need to get out of the terminal cisterna. We need to eventually get it into the sarcoplasm. More on that in a little bit. So here in the sarcoplasmic reticulum, we've got calcium pumps. And so we know what those things do. They're going to move things against the concentration gradient. In this case, our calcium pumps are going to take calcium that's in the sarcoplasm and dump it into the sarcoplasmic reticulum. Okay, that's pretty much, think of these calcium pumps as the crew that comes in to clean up after a party, after New Year's Eve in Times Square. Okay, so we just had a party, and our party was, we had a muscle contraction going on, and now we're done contracting, but we have all this calcium that's lying around in the sarcoplasm. It's floating around there. We got to get rid of it. We got to clean that up. So the calcium pumps do that. So they pump that calcium out of the sarcoplasm and into the sarcoplasmic reticulum, where we have two calcium storage proteins just waiting to bind onto calcium to store it for a while. So those two proteins are called calmodulin and calsequestrin. Easy to remember calsequestrin because when you sequester a jury, you're going to put them in a room somewhere. Well, in this case, calsequestrin is a storage protein for calcium that's going to bind calcium and keep it in the terminal cisterna. And it just waits. Okay, and it waits to be released. Well, what's going to release it? These fellas right here, the calcium release channels. Once that electrical signal comes down the T-tubule and triggers the opening, all right, via the voltage sensitive calcium channels, it's going to trigger these calcium release channels to open up and release calcium back into the sarcoplasm so we can have another muscle party. And then the whole process gets repeated. After we're done contracting, then those calcium pumps pump the calcium out of the sarcoplasm back into the terminal cisterna, right, in the sarcoplasmic reticulum. And then we wait until the next muscle party. So this is what I'm talking about. Here's our T-tubule, right? So our electrical signal is going to go down the drain, right, of our bathtub here, goes down here, and it triggers the opening of these voltage sensitive calcium channels. And when those voltage gated calcium channels open up, right, that allows this calcium release channel to let calcium leave the terminal cisterna and go right back into the sarcoplasm. Because that's what we want to do. We want to get that calcium into the sarcoplasm so we can get a muscle contraction. Once the party's over, these guys here, the calcium ion pumps, bring calcium back into the terminal cisterna. And so you'll have free calcium floating around in here, but we'll also bind some of that calcium to calsequestrin and calmodulin. It's not uncommon. I'll talk more about this. Okay, so it's really important that you kind of understand that in regards to what the role of calcium is. You know, again, if we don't have it, we cannot have a muscular contraction. So now we're going to enter in and discuss some of the players, all right, or the things that are going to be involved in this muscle contraction. So I have to start off by talking about myofilaments, right? We learned about these structures before when we were talking about the cytoskeleton, and we learned about the different types of cytoskeleton filaments here. And so our myofilaments are contractile proteins. That's important that you know that term, contractile proteins. These are going to be the ones that are going to interact with each other. These protein filaments are going to interact with each other and cause the muscle to contract, change shape. So there's two types. We have a thick filament known as myosin, and then we have a thin filament called actin. Okay, so we have a thick and thin filament, myosin and actin. Usually it's actin myosin. Right, so let's just talk about myosin here for a moment. This is our thick filament. Myosin has these heads, these globular heads. And they're going to be on either end or point towards either end of our filament here. And so on this head, and I'll try to draw it as best as I can. All right, it'll have a tail that comes off the end here and runs like that. That's actually not too bad. Okay, so here's the myosin head, and then here's our tail. Now the head has a couple of binding sites, 2 binding sites on it, 1 and 2. One of those binding sites is for our thin filament, actin, so we call that the actin binding site. The other one is going to be the ATP binding site. Okay, well, we need ATP. ATP is our energy currency, so it makes sense. So we have one binding site for actin. The other binding site is for our ATP. Unfortunately, well, I shouldn't say unfortunately, it's a good thing, but when ATP binds onto that site, it's eventually going to split into one ADP molecule and one phosphate molecule. And we'll talk you through why that happens. All right, so our other contractile protein is the thin filament. The thin filament, I want you to think like a pearl necklace, okay? We have, right, I can't really describe it much better than a pearl neck, like 2 pearl necklaces, and we just twisted them around one another. And so there's 2 types of strands here. One of those strands is known as the F-actin. The other strand is known as the G-actin. And so it's the G-actin that's going to have our myosin binding site. Well, that's good because remember, we're going to have these 2 contractile proteins interact with each other, and then they're going to change the shape of the muscle. Okay, so the G-actin has the myosin binding site, and that's where the myosin is going to actually attach onto when we start to shorten the muscle. And then we have 2 other proteins that are definitely worth knowing, tropomyosin and troponin. These are categorized as regulatory proteins. All right, it's these proteins that are going to regulate whether or not we have a contraction or not. Because when the muscle's at rest, you have tropomyosin that sits on top of the myosin binding site on our G-actin. So it sits there. So if it's sitting on top of the binding site, that means myosin cannot bind to actin. That means we can't have a contraction. So that's a problem. So we got to get that tropomyosin off of the myosin binding site so we can have a contraction. Well, we got another protein here called troponin, and that sits on top of the tropomyosin. And it's got a receptor. And its receptor is for, yes, you guessed it, none other than calcium. And so when calcium enters into the sarcoplasm, because that's where these filaments are, these protein filaments sit inside of that sarcoplasm. So when calcium enters into the sarcoplasm, it binds on to the receptor binding site there on troponin. And that triggers troponin and tropomyosin to move off of the myosin binding site so we can have myosin and actin bind to one another. And so that's what we're seeing here. Okay, so here's our thin filament, our actin. So you got your G-actin and your F-actin. As you can see, it looks like 2 pearl necklaces that were just twisted around each other. So on the G-actin, that's where we have our myosin binding site, but it's blocked by tropomyosin. It's a rope-like regulatory protein. And then what sits on top of that is the troponin. It's a little cup-like structure. So what will happen is calcium will come on. It'll sit on the calcium binding site of troponin. That triggers the movement of troponin and tropomyosin to shift away from the myosin binding site. That myosin binding site now is exposed, and that allows our thick filament here, myosin, to attach onto the actin with one of the globular heads here. And that's what we'll see. So this picture here is actually showing you, let me zoom back in prematurely. All right, so here's our muscle fiber, that's our muscle cell. And it's made up of these myofibrils, these long tube-like structures. So we slide that out, and then inside of these myofibrils, you'll have the myofilaments. Myofilaments are those contractile proteins. I can't zoom in anymore. Here you can see, right, those myofilaments on a larger scale here. So those myofilaments are these, the thick and thin filaments. Okay. So again, if you're not quite sure, go ahead and review that in your book. Okay. So what else do we see in our muscles? Well, I should be more specific. What else do we see inside the muscle cell? Well, we have the mitochondria. Okay, the mitochondria is going to be the structure, okay, or the organelle that is going to make our ATP. And it does so through aerobic cellular respiration, which is the most effective form of energy production, meaning you get the most bang for your buck. All right. For example, one glucose molecule utilizing aerobic cellular respiration will give us 32 ATP. If we're not utilizing aerobic cellular respiration, one glucose molecule gives us about 2 ATP. So we want to use aerobic cellular respiration. So therefore, this tissue has lots of mitochondria in it. Also, we'll see in skeletal muscle a protein called myoglobulin. We've heard of hemoglobin. Okay, hemoglobin is the oxygen carrying molecule inside of our red blood cells. Well, myoglobin is going to carry the oxygen inside of our cells so we can utilize that oxygen for our cellular aerobic respiration. Right, aerobic means it's a nice way of saying we need oxygen. If you don't have oxygen, you cannot undergo aerobic cellular respiration. So if we have oxygen, we're going to use it. If we don't have it, then we're going to undergo anaerobic cellular respiration, and that's a problem. So in this scenario, we got to keep on hand a protein that's going to store oxygen. So our muscle cells have this myoglobin, and that allows the mitochondria to have oxygen on hand. We'll also need something to help make ATP. Well, gosh darn it, there's a glycogen. What's glycogen? That's the storage form of glucose. We talked about that before. So these muscle cells, there's only two types of cells or organs that are going to store glucose. That's going to be the liver and muscle, okay, specifically skeletal muscle. So we have glycogen, which is the stored form of glucose. And then we also have this molecule called creatine phosphate. And creatine phosphate is going to be this molecule that is going to help us to replenish our ATP supply. We use a creatine molecule that carries a phosphate attached to it. So when we break ATP down, because we're using it for energy, we get left with ADP and one inorganic phosphate molecule. Well, what creatine phosphate does is it gives up its phosphate molecule so we can recycle that ADP into ATP. So it helps us to replenish that ATP supply. Very, very nice, very, very selfless, kind of like that. Okay, so we're only going to find myoglobulin in muscle tissue. And that's what gives our muscle tissue that reddish pigmentation. So when the muscles aren't doing anything, that myoglobulin, goes around and it picks up oxygen. So it's ready, all right, for the next time that you have to engage in that aerobic cellular respiration. So it keeps that right on hand. Okay, so now we know some of the players that are involved. Now we're going to talk about how we are going to stimulate and create a skeletal muscle contraction. And so there's going to be three events that we're going to discuss. The first one is called the neuromuscular junction event, in which we have excitation of the skeletal muscle fiber. All right, the next one is going to be the excitation contraction coupling. And the third one is going to be the cross bridge cycling. So we're going to talk about each of these events kind of like a story. So we're going to start off by what happens at the neuromuscular junction. That's going to be where our motor neuron is going to synapse onto our skeletal muscle cell. So let's discuss that. All right, so first of all, what is a neuromuscular junction? That's where a motor neuron is going to synapse right in the middle of our muscle fiber. And this is where we're going to see the synaptic knobs hovering right above that muscle fiber. So that involves three structures: the synaptic knob, which comes from the motor neuron, the motor endplate, which is that area in which the synaptic knob is going to approximate on the muscle fiber, and then the space in between, which is called the synaptic cleft, which we are familiar with. So let's review what a synaptic knob is. All right. At the terminal end of our neuron, right, we have these swellings, and those are the synaptic knobs. They are considered to be the transmissive segment of our neuron. That's the last segment of our neuron. Inside the synaptic knob is where we store our neurotransmitter, which is that chemical which is going to influence some sort of effect on the effector organ or whatever the other, or another neuron. So that neurotransmitter is stored in these little bubbles called synaptic vesicles. And in this case, since we're talking about skeletal muscle, the neurotransmitter that we're going to use, we're going to give a name now. So finally, we can put a name to this neurotransmitter that we've been talking about. And there's many types of neurotransmitters, but since we're talking about skeletal muscle, let's talk about acetylcholine, ACH. Acetylcholine is going to be the neurotransmitter that is going to be used to stimulate our muscle tissue. All right, we're also going to see in the synaptic knob, we saw this before, our calcium pumps, because what is their job? To pump calcium out of the cell, creating that concentration gradient. Because then by doing that, calcium wants to enter into the cell. So once that action potential reaches the synaptic knob, it's going to trigger the opening of voltage-gated calcium channels. Right, so once that occurs, those voltage-gated calcium channels open up. Calcium then diffuses down its concentration gradient, so it moves from the outside of the cell to the inside of the cell. And then what it does is it binds onto the surface of the synaptic vesicles. And if you notice, calcium, where are you? Calcium is a cation. It's positively charged. So it coats these synaptic vesicles, which is nice, because now it's made those synaptic vesicles polarized. And so now those synaptic vesicles have a positive charge. Well, if you remember, the inside of the plasma membrane has a negative charge. So those positively charged synaptic vesicles are attracted to the plasma membrane, which is good. They're attracted to the plasma membrane, they fuse with the plasma membrane, and they release, all right, the acetylcholine through exocytosis. You guys remember that? Exocytosis. All right, so it releases the neurotransmitter through exocytosis, and we get all this neurotransmitter flooding into the synaptic cleft. And that's what this picture here is showing us, okay? That acetylcholine diffuses across the cleft, and then, I'll zoom in here, and then it binds onto, right, the receptors on our muscle. So here you can see, here's our neurotransmitter, the acetylcholine gets released into the synaptic cleft. It diffuses across the synaptic cleft, and it binds onto the receptors, right, for these transport proteins right here. And what that will do is that will cause the triggering of the opening of these channels, and then we can get ions to diffuse across the plasma membrane there. All right, so this is what we're going to see right, at the neuromuscular junction. So now you get to see it in effect. All right, so we're going to discuss how that occurs. But this is what happens. This is the first physiological event, all right, when we're talking about the excitation of the skeletal muscle fiber. Okay, so the acetylcholine diffuses across, all right, the synaptic cleft, and it binds onto the receptors here on these channels. Now, where the synaptic knob, okay, comes in contact or hovers above our muscle fiber, we call that the motor end plate. Okay, the motor end plate. So we have these different channels, these different receptors on the motor end plate that acetylcholine has now bound itself to, which takes us into our second physiological event, which is the excitation contraction coupling. So we haven't really quite created our own electrical charge, because that's what we're trying to do. We're trying to create an electrical charge on the sarcolemma. That's the plasma membrane of our muscle cell. So this is how we're going to end up doing this. Okay, because remember, our main goal, believe it or not, all of this is in an effort to release calcium from the terminal cisterna. We got to rescue our princess. So we're going to go to all this trouble to release calcium into the sarcoplasm of our cell. Because right now it's not in there, it's separated, right? So we're going to see now how this occurs. So once we undergo this process, and I'll walk you through all these steps that are listed below, once we release that calcium, then we can move those regulatory proteins off the binding sites of our myofilaments, of the actin and myosin, and we can get what's called a cross bridge, and we can undergo muscle contraction, and that's our goal. So our end goal, all right, was to cause a muscle contraction, but we want to release calcium that's imprisoned in our sarcoplasmic reticulum. Right, so here's what happens. I'm jumping ahead here. All right, let me zoom in so you can see what I'm talking about. Okay, so we've released, all right, our acetylcholine into the synaptic cleft. It's triggered the opening here, all right, of these channels here, which allows the diffusion, all right, of our ions across the plasma membrane. Our goal here is going to be to change the membrane potential. Remember membrane potentials, that's that electrical charge across the plasma membrane. Our goal is to change the membrane potential here. Now, if you recall, the resting membrane potential in a neuron is negative 70 millivolts. Our threshold value was negative 55 millivolts. Well, here in the muscle cell, it's a little bit different, right? First of all, we don't call it the resting membrane potential. We call it the end plate potential. And so what we are going to see is we want to create this end plate potential, also known as an EPP. And we need to, again, get to that threshold value, right, which is going to be, in this case, a different value than what we saw in the neuron. All right, so our goal is to generate this EPP, which will eventually trigger an action potential. So we open these gates up. We have sodium gates being opened up and potassium gates being opened up. That allows sodium to enter into the cell and potassium to leave the cell. All right, but what we'll see is, we'll see the sodium flows in, okay? And what will happen is we will change our membrane potential from that negative 90 millivolts, and our goal is to get it to that threshold value, in this case, negative 65 millivolts. So that's a total of a positive 35 millivolt difference. So when we open up these channels here, you're going to see, right, you're thinking probably, hey, wait, Dr. Kaz, potassium's leaving the inside of our cell, and so won't that make the inside of the cell, all right, more negative? Yeah, all right, but way more sodium enters in. Okay, sodium, all right, is going to move down its concentration gradient, but also it's going to get pulled in through the electrical gradient because it's negative inside the cell. So sodium pours in way more, all right, than potassium leaving the cell. So that's going to drop our end plate potential from negative 90 millivolts to negative 65. And so when that gets to, like we saw in our neurons, when it gets to a voltage gated sodium channel, boom, that's when we get our action potential. All right, remember that all or nothing scenario? Okay, so when we develop our end plate potential, right, we reach our threshold value. Okay, we start to depolarize the plasma membrane here. It reaches its first voltage gated sodium channel. And then that voltage gated sodium channel, all right, opens up, and that allows sodium to enter into the cell, and it causes depolarization of the cell. And so we're able to generate that action potential. In the same scenario, right, we have sodium, we have voltage gated sodium channels, and we have our voltage gated potassium channels, because, right, what goes up must come down. We're going to depolarize the cell, and then we're going to follow with repolarizing the cell. So that's what we're going to see here. And so this is going to continue all the way down the plasma membrane until we reach a T-tubule. So we generated our action potential way over here off screen, and our action potential starts flooding down the sarcolemma. And there's thousands of T-tubules all over the place. All right. So our action potential finds a T-tubule, goes down into the T-tubule, and then it'll start to encounter these voltage sensitive calcium channels. And it'll trigger these voltage gated calcium channels to open up the calcium release channels, right, that are found on our terminal cisterna. And so remember, our terminal cisterna is that tower that our calcium princess is trapped in. And so now calcium can flow out of the terminal cisterna into the sarcoplasm. Because what's waiting in the sarcoplasm? Our myofilaments, our actin and myosin myofilaments that are going to need calcium to remove those regulatory proteins so they can start to interact and start a contraction. So that's what we're seeing here. All right, we're going to see this develop here in a moment when we talk about our cross-bridge cycling here in a moment. And then once the party's over, all right, then our calcium pumps will pump calcium out of the sarcoplasm and back into the terminal cisterna there. All right, so this slide here shows you, all right, all the events. I'm telling you, if you know this slide, all right, then you can learn more of the detail. 4 simple events that occur, all right, for an action potential at the sarcolemma. All right, so first our muscle is just minding its own business. It's just resting, all right? So it has its resting membrane potential of negative 90. Okay, now our motor end plate, all right, starts to depolarize because the neurotransmitter there from our motor neuron has triggered the opening of those voltage gated channels, or excuse me, not voltage gated channels, our chemically gated channels there. And so we see an opening of our chemically gated sodium and potassium channels. And so we'll start to see depolarization occur. And once we get to that threshold value of negative 65, okay, as that occurs, then we hit that first voltage gated sodium channel on the sarcoplasm, on the sarcolemma, then it's game on. All right, that opens up and it's the domino effect. So the cell will depolarize, going to that positive 30 value. Then our voltage gated sodium channels will close, and then our voltage gated potassium channels will open up and will repolarize the cell. The same course of events will happen. It's real basic here. All right, so that's what we're seeing here. All right, I mentioned this before. The action potential will travel down the T-tubule. And then the T-tubule will trigger the opening of the calcium release channels, and that will allow calcium to move out of the sarcoplasmic reticulum and the terminal cisterna out into the sarcoplasm. And then that's where our calcium will interact with those regulatory proteins and cause those myofilaments to interact with one another for contraction. And so we get to talk about that next, right? Because this is the last physiological event, all right, of muscle contraction, which is the cross bridge cycle. All right, so we've released calcium. The princess is out of the castle, okay? She's going into the sarcoplasm and she's looking for troponin. Okay, that cup-like protein that sits on top of our tropomyosin, which is that rope-like protein that blocks, all right, the myosin binding sites on our G-actin. So once calcium binds onto troponin, it moves all of that away. And so now the actin is exposed. The myosin binding site on the actin is exposed. So that will allow myosin to bind onto the actin. And that's what we're going to talk about now. Okay, we're going to discuss that. All right, so I've already talked about the calcium binding. Calcium binds onto troponin. Troponin then causes the tropomyosin complex to move away from the myosin binding sites. And now the myosin binding sites of actin are exposed. So let's talk about our cross bridge cycle. There's going to be 4 steps here. So I'm going to go over one step, show you a picture, then come back. The first one is the cross bridge formation. So we haven't actually made contact between the actin and myosin here. So in the cross bridge formation, those myosin heads that we were talking about, these guys down here, these heads are in a cocked, what we call a cocked position. All right, if you've ever seen in the movies, some guns have that little, they call it the hammer, where you'll pull it back on the revolver and they'll pull it back into the cocked position. That's what we'll see here, all right, with our myosin heads. They're in this cocked position and they are ready to attach onto the actin. And so this is what we're going to see here. So let's show you. All right, so this is our cross bridge formation. This is where they attach. So we're in the cocked position. We've just exposed, all right, the myosin binding sites on the actin. So the myosin head will extend and contact that binding site, creating a connection here. And we call that the cross bridge. Okay, that's where they attach. That's the cross bridge. All right, so that's step 1. Step 2 is what we call the power stroke. This is when we're actually going to see movement. Right, so what will happen is that myosin head that was cocked will now swivel. And as it's swiveling and moving, it's going to pull the actin filament, the thin filament, past the thick filaments. As this occurs, remember I told you we have 2 binding sites on the myosin head. One is for actin, the other one is for ATP. Okay. At this point, that ATP has undergone a chemical reaction, and it is ADP and an inorganic phosphate. So both of those molecules were attached onto the ATP binding site. So when the power stroke occurs, both of those molecules pop off. And that allows that binding site to be available for another ATP molecule to bind on. So this is what we're seeing here. I didn't address this up here, but here you can see our binding site for the actin is now covered because we've connected to the actin, but we still have our ADP and inorganic phosphate molecule. So what will happen is this head will swivel. Okay, you can see how it's at, kind of off to the right here. Well, when we undergo the power stroke, you can see it pulls things here to the left. And as it does that, okay, as it does that, ADP and that inorganic phosphate will pop off, right, exposing the ATP binding site again. That's a good thing. Okay, so we're still connected. We've done the power stroke. We pulled everything by, right? Maybe we want to do it again. Maybe we want to have another power stroke. We want to contract our muscle further. Or maybe we just want to stop this whole thing, right? Well, we have to release this connection here. Well, that's our third step. Right? That is where we release the myosin head from the actin. In order to do that, guess what? We need ATP. So ATP comes bounding along. It attaches onto the ATP binding site. And as it does so, that myosin head pops off of the actin. That's our release phase. Okay, so here you can see, all right, the myosin head popping off of the actin binding site. As ATP binds onto there, it causes, all right, myosin to pop off. And then finally, our last step is we have to start this process over again, but to do so, we have to reset or recock that myosin head. As we do that, ATP hydrolyzes or breaks down into ADP and that inorganic phosphate. The enzyme that is responsible for this reaction is called ATPase. ATPase, any word that has ASE on it, is an enzyme. So ATPase is going to be the enzyme that breaks ATP down into ADP and inorganic phosphate. That's what that little I means behind the P. And so that allows us to recock the head. And that's what we see here in this picture. Okay, so now when it breaks down into ADP and inorganic phosphate, we've recocked the head. And then we will recycle the whole thing here, right, for another contraction if need be. So you folks now know, all right, cross bridge cycling. So what is the function of calcium in skeletal muscle contraction? Remember, calcium is that princess. We need her, we need calcium to bind on to troponin, which will cause that conformational change in the troponin-tropomyosin complex, which blocks, all right, the binding sites for myosin and actin. Okay, so once calcium binds on troponin, it shifts the troponin-tropomyosin complex off of the binding sites of actin. So now myosin can bind, and so we can have cross-bridge cycling. So what causes the release of the myosin head from actin? Right? ATP. ATP is going to cause the release of myosin, or cause the release of the myosin head from actin. So if you're not generating ATP, okay, your muscles cannot relax. They can over time. That's what rigor mortis is. And when you're dead, you're not making any more ATP because you're dead. Rigor mortis is only temporary because, all right, those proteins start to break down and degrade over time. So what resets the myosin head, right? The resetting of the myosin head is going to be that ATPase enzyme breaking down ATP into ADP and the inorganic phosphate. Okay, I think I'm going to stop here for today.